Hi, this is Dr. Yunong Liu from Department of Medical and Molecular Genetics. In this short module, we're going to go through a file format called VCF files. Briefly, that standard NGS format include the FASTQ files, SAM files, and VCF files. And VCF files is the last part that represent the variant information. VCF is also called the variant call format, and it is standard format for storing the most prevalent type of sequence variation, including SNPs, indels, large structure variations, together with very rich annotation. And it is also in a very compressed manner so that it can save the space and it can be indexed. So some of the information is easier to be retrieved. Similar as the BAM file we talked about before, and VCF file has actually two components. The first one is the header information that include a lot of information regarding the variant calling that being used in this particular, through the generation of this particular VCF file. And the body part here is the one that really tell us where this particular variant is. So we just directly dive into this body part of it. I will go through a few concepts. Most of them are very self-explanatory. So for example, initially it's a chromosome number, and in this case it's chromosome 20, and position is 14370, and the ID is whether it has a specific RS number, which means that whether this particular variant, we have seen that before, has been already documented into the database. Normally when it's a dot, that means we have not seen that variant before. And reference and alternative, and this is going to be the nucleotide on the reference genome and the nucleotide on the alternative genome. So let's take on this particular donor reference. So let's take a look at this. When we talk about the SNPs, and then this is very, very straightforward. So you can see this, the top one is the reference, the bottom one is the donor, this particular individual, what the sequence is. And this is position number 2 here, and the reference is C, and the alternative is T. Very straightforward. Next one is the insertion. So you can see that in the reference genome is ACGT, but there's one more nucleotide that is in this particular donor genome. And then you can see it tells you this is the insertion here. Well, when we document the position, we will document the nucleotide, the base that before insertion happened, which is position number 2. And then the reference is C, and the alternative is the CT. So this tells you this is a particular insertion. For the deletion, and then you can see this, the rules start to change a little bit. For example, this reference genome is ACGT. In the donor sequence is A and T. So C and G are being deleted. And the position we're going to record is going to be the base that before that happened, which is position number 1. In the reference is ACG, in the alternative allele, it is A only. And there will be more complex events, so I'm not going to go too much detail because this doesn't happen very frequent. But in this case, you can see that this reference is ACGT, and in the alternative in this donor allele is ATT. So the position you still put it in the number 1 here, and the reference is ACG, and the alternative is AT. So this is a more complex events. And also it can record the larger structure variations, which I'm not going to go too much detail about. The next field is the quality information about this variant. And in this case, that all the variant quality is still defined as the Fred quality score that we mentioned earlier when we discussed about the FASTQ files. For example, the quality is always the minus 10 times log 10 of the probability you are making mistake. And in this case, remember we talked about if it's 30, that means the probability, if Q is equal to 30, that means this probability is equal to 0.001. That means that if I say there is a variant in this location, I will have 0.1% of the chance making mistake. So again, the smaller the p-value is, the better the quality is, the more accurate our variant calling is, and the larger the Q value is. Just keep that in mind. And the field afterwards is a filter. So you can see that most of them are said pass. So what that really means is it pass all the filters being defined earlier. Where these filters are being defined, they're actually being defined in the headers. So remember that we talk about the body of the file and the header. In the header, there could be some of the lines that defines this filter rules. For example, the filter is equal to, this ID is Q10, that means description is quality is below 10, and then there's another filter being defined here, is less than 50% of the samples have that data. So in this case, that you can see in this particular one, the second row here, and they didn't say pass, instead it's put the Q10 there. That means this particular filter are being violated, and it's the quality of this particular variant calling is less than 10. So we need to pay attention to that. And sometimes that you will have situations that multiple filters are being violated. So let's go to the next field, which is the information. So this is the information for this particular variant location. So you can see there are many defined areas, and let's look at there are 18 predefined options. Again, this is something that is constantly evolving. For example, if it says DB, and that means whether this is in the DB SNP membership, if it's DB, so you can see that this particular one, we already have an RS number, so this is being known before. And DP is combined depths across all the samples. And then, so in this case, there will be 14 sequencing rays that map against on this variant location, so that help us to call the variants. So the sequencing depths, normally the higher the sequencing depths, and the better off we are, the more evidence we have to call this as evidence, this a variant or not. NS, the number of samples with data on this particular location, and AF is estimated allele frequency, and so on and so forth. So I'm not going to go detail of every single one of it. But the general point I want to mention here is this is the field that tells you the information about this variant loci. Okay, so just to keep in mind, and then we will start to go into these areas, and then we can potentially loop back to this, and hopefully that'll make better sense here. So now let's come to this particular sections, which tells you the individual genomic variation information. So the concept here is you don't have to have just one VCF file just for one individual. As a matter of fact, one VCF file can document the genomic locations of many individuals. In this particular example, there are 3 individuals here. And actually in the 1,000 Genomes project, there are over 2,500 individuals. Their sequence are available. There is only one VCF file that includes all the 2,500 individuals. So you can see that each block here corresponding to one particular individual. So within this, you will see all those different numbers here. And what this number really means are being defined in this format field. So let's go into a little bit more detail and to see what this really means. The first one is GT here. In this case, is the GT. The GT stands for genotyping information. So if you see this is 00, that means this is not an alternative allele. So for this individual, the sequence, the nucleotide will be the same as the reference genome. In this case, the reference genome is G. So both two alleles of this individual will be G allele, it's GG. Well, for the second individual, so you are seeing here is 1, 0. And that means one allele is the same as the reference and the other allele is different. It's the same as alternative. So in this individual, at this first particular genomic location, it is a heterozygous variant, and then the allele will be G and A. Both two alleles are being present. Well, for the third one here is 1, 1. That means it's a homozygous alternative allele. So both two alleles are the alternative allele A. So that is the first field. What that means is defined by this header information in the format, which is GT, that stands for the genotype. And the next field here is GQ. So what GQ means is a conditional genotype quality of this particular individual. As I mentioned earlier, this is a Phred score calculation. The larger the number is, the better off we are. The next one is DP, is the depths, the read depths at this position in this particular sample. So you can see this is 1, 8, or 5, okay? 8 plus 5 is 13, plus 1 is 14. So collectively, across 3 individuals, the overall sequencing depth is 14. So now let's go back to here that define the entire of all the samples in this particular genomic loci, and that is very clear that this is a 14. And the HQ is a haplotype quality and things like that. There are many, many different fields are available in this part. Okay, so now this is clear that this particular section are defining the variations for each individual variance, in each individual people that are being included in this VCF file. So, and now coming back to this section instead is the definition or some of the characteristics of all the individuals in this particular genomic location. Well, so if you think those are not complex enough, actually VCF can be very, very complicated. It can document many different types of genetic variations. In this case, it is in the cancer genome, we know there's a lot of structural variations are available, and then actually they can be flexible enough to include the information like this type of structural variation. Or you think this is still not complex enough, actually this is possible too. Honestly, I don't know how to do that, and unless you are doing the structural variation research every day, and you probably don't need to know the details, but just have a general idea, all these informations can be documented in the VCF format. So that's all I want to talk about today, and I hope you enjoy this module. And from next week, we will start to talk about RNA sequencing data analysis.